Thank you. I first would like to thank the organizers to inviting me to this meeting. It's been very exciting. And I guess I'm going to be continue the same theme, different channel, what Cryo-EM could tell us about mechanisms and functions of the specific ion channels. In my lab for 20 years almost, we studied transit receptor potential channels, and I don't really have to introduce you guys those channels. Everybody know that they're very exciting and very different from some other channels. And we are really all studying them because we're partially trying to understand what of those 28 homologs do in a body, and they're involved in many physiological and pathophysiological processes. But my lab specifically focus on TRPV channels, and of course, they are probably one of, I call the best channels to study, because we all originally was chasing how they're involved in pain. But now I think we're trying to more and more understand what exactly do they do in humans. So TRPV channels is divided in sort of two subfamilies. TRPV1 to TRPV4 represent those non-thermo TRPV channels that are non-selective cation channels and express widely everywhere, but originally, like I said, the focus was on neuronal systems. And it's another two channels, TRPV5 and TRPV6, that are very similar to each other, sequence-wise, while the TRPV1 and TRPV4 are not. And they're also very highly calcium selective. So one channel, TRPV6, only expressed in intestine, and another one is only expressed in the kidney. And they involved, one is in absorption and one is in reabsorption of calcium back into the system. So if I have time, I will tell you first about the TRPV5 channel that we study. And if we really have time, I will tell you about TRPV2. So TRPV5 is a very selective calcium channel, only expresses in the kidney, as I said, and really responsible only on the 10% of the reabsorption of the calcium back to the blood. But if you have mutations in this channel, what happens is that as the calcium does not get reabsorbed back, and you form a lot of calcium stones, and that is very painful disorder. So we also decide to study this channel not only from the point of view of understanding how it works in the kidney, but sort of like a channel that very specifically expressed in one specific tissue, try to understand what can we learn about the gating of this channel using Cryo-EM. So the most interesting part of about this channel is this only expressed in apical membrane of the epithelial cells. It actually sits in a very interesting lipid composition in the membrane. It has cholesterol, phospholipid, and glycolipid 1 to 1. It's really modulated endogenously by PIP2, and it has other molecules that interact with the channel to inactivate it. An example is comedulin, or activated at pKa. And also, pretty much every hour your pH in the urine change, so pH 4.5 to 8 constantly modulate this channel. So as been already told here is that Cryo-EM is an amazing technique that could give you any information that you would like. And so we started first from trying to understand the endogenous modulation by the lipids. So we work only on the channels that are wild type. We reconstitute them into nanodiscs, so they constantly sort of represented in the membrane-like environment. So the first structure we solved was pretty much lipid-bound state, so what we call it apo state. The structure revealed a lot of bound lipids, and it's also revealed the pore of the channel, where this is the upper gate and it's a lower gate, was partially open on the bottom, but the calcium could not go through the channel. So well, that all makes sense. So we were not really satisfied because we wanted to see the channel open. So we add the PIP2 to the mixture into the nanodiscs, and what we noticed that we clearly could resolve the structure of the PIP2, and it was really coordinated by the charge residues located between S6 and S4, S5 linker of the channel. So those arginines and lysines were coordinating this lipid very well, that when we mutated and compared to actually of the predicted molecular dynamics model of the PIP2 interaction with the other channel, TRPV6, this pretty much fall into the places that mutating those residues completely abolish interaction with the lipid. So we also noticed that the pore was widely open, so now you could see the calcium could clearly flow through the channel, what was very satisfying to see the channel in both conformations. And here we could see that the upper gate, the aspartic acid 542 was coordinating the calcium in the upper pore, and on the bottom, the tryptophan 583 was also moving out of the way, allowing the calcium to go through. So this was all possible, again, through the conformation and interaction with those charged residues. And here is just the movie. It is just a combination of open and closed state that we solve. So this is not really true. It's just a cartoon. But here it just shows you that now if you have PEEP2, what's going to appear in a second, you could open the channel and the calcium could go inside of the cell. Of course, cell cannot tolerate that much calcium going inside all the time. So the next question we ask using Cryo-EM, can we figure out the way how this channel gets inactivated? And of course, we knew that the channel could be inactivated by comodulin, and tried to figure out how this happened at the molecular level. Again, without Cryo-EM, probably wouldn't be possible. So we used comodulin, and we basically based our knowledge about the inactivation of the channels by the comodulin is that it was an interesting cartoon published in 2013, but one of my collaborators suggesting that while PEEP2 does activate the channel, calcium goes through, somehow calcium binds to the comodulin, and comodulin sort of blocks the channel. So this was just a cartoon representation, and we didn't really know much about it. We had some structures of the small little peptides from the comodulin bound to the channels, against to the small pieces of the channels. But really, the proposed model was it's one or two comodulin somehow interact with the channel and allow the calcium to basically no more going inside of the cell. So again, Cryo-EM makes the wonders. You take the proteins, you basically incubate this with the inactivated molecule like comodulin, and to our surprise, we saw one comodulin bound inside of basically the skirt of the anchoring repeats, and we were able to resolve the whole comodulin in a calcium-bound state, and we also saw some peptides, the same as an NMR, of the C-terminus of the channel. They were interactive with the C-lobe and N-lobe of the comodulin. Surprisingly, we were not able to figure out which part of the full C-term interacts with the comodulin, but it was clear that C-terminus was bringing the comodulin closer to the pore. So again, the pore was closed, a little bit tighter, and the interesting part was from all the study was this lysine 116 on the C-lobe of the comodulin was literally going inside of the cell and sort of almost like a cork plugging the bottle and sitting and not allowing their, coordinated by those 4 tryptophans on the bottom of the pore, not allowing calcium flow anymore inside of the cell. So this mechanism is quite interesting. So the comodulin basically comes in and plugs the pore. And again, it's only one comodulin that we see that interact directly with the channel. It's possible that one of the another second comodulin is always also attached to the C-terminus, but it's hard to show that in the dynamic system when we're solving the structures of the protein that's quite dynamic. So we also mutate that tryptophan, and here we could abolish all the comodulin inactivation. Seeing here is that if we have PEEP2 and we have the mutant that does not have the tryptophan, calcium is still continue flowing inside of the cell. So this was all nice and interesting because we were able to see endogenous modulators. We wanted to ask a question now about the drug discoveries and look at the endogenous modulators of the channel. So the TRP channels is notorious known that we don't have really very specific activators or, inhibitors. Maybe only one good example is a capsaicin and TRPV1. So iconazole sort of a very dirty inhibitor of the TRPV5, and it's an antifungal cream. So it's not that easy to put this compound in the solution. And it's a recent paper about the tetrahydrocannabinol divarine that has been shown to in the channel also. So we solved the structures of both here. Just I'm going to show you the iconazole structure. And the vanilloid pocket in TRP channels is known to be occupied by vanilloids as in TRPV1 and some of the compounds or lipids in this pocket. So we solved the structure with iconazole and saw that the compound possibly sits in the lipid binding site. To our surprise, also when we were solving the structure of the APOD channel in detergent or in nanodiscs, we also saw that the lipid actually sits very well in the same binding pocket. So we used in silico screening what is basically using the zinc 15 compound library that you could buy in 15 minutes from the company in Ukraine, and they ship it to you in 10 days, and you basically could get all the compounds that you find through the in silico screening by using structure-based drug design. So those are compounds that didn't work out, but the ones that work out was quite surprising. So we first found the parent compound, and this is just going to be the number here, 5420, that was actually inhibiting both TRPV5 and TRPV6. But when we asked the company to send us all the derivatives that they had, and they had 10 of them, one of those derivatives, 8990, was completely inhibiting TRPV5, but was doing nothing to TRPV6. That's puzzled us for so long because we repeated this experiment many times, because V5 and V6 is 99% sequence homologous. They do the same work. That was very surprising. So of course, Cryo-EM again could do the wonders. You have your compounds, you put them on the proteins, and start solve the structures. And a Cryo-EM also could reveal an interesting story. So when we used the original compound, we saw that the compound actually binds between two lipids, that one of those lipids still occupied this vanilloid binding site, and another lipid coordinated closer to the pore. So this binding site was also occupied by the parent compound, but the other binding site got revealed in a completely unexpected position. So what's interesting about the study is that while we were expecting and modeling everything in our binding site weights would be for the lipids and aconazole, we actually find out that those lipids is very hard to exchange. The drug actually was binding in a completely different site, and when we reran our drug screen, assuming that the lipid is still sitting in that binding site, we actually found those compounds again being one of the top hits. So when we looked closely into the binding site for the compound, the derivative compound, the one that was very specific for TRPV5, we noticed that actually binds in this S4-S5 bundle of the channel. And when we mutate the position where it was sitting, we noticed that again we could manipulate and see that this binding pocket probably valid. But what's more interesting part is to try to figure out what the mechanism and why is it so different from V6 and how does it really inhibits the channel. So you could see here this is the PIP2 structure that I already described of the open channel. This is the lipid structure where we have pre-open channel. And this is the structure with the inhibitor. Pore is completely closed. And so we start thinking how does it all possible and how the inhibitor works. In the pink is here is the lipid bound structure. In the dark red, I guess, would be the inhibitor bound structure. And you could see very little movement in this position where the channel, when the drug is binding. When we compare this structure to the point where the green would be your PIP activated structure, so fully open channel, the drug is wedges itself enough there that if we apply PIP, it would not allow to open the channel. So basically we're inhibiting the site in a channel where this inhibition preventing the movement of TRP box and the S4-S5 linker and not allowing at all of PIP2 activation. So based on this study, we could say that Cryo-EM allowed us to show inactivation by comedulin, activation by PIP2. We have several drug structures, and from this drug structure of the last compound that we found, it's actually quite fascinating that it's binding in a completely different site, but it's affecting the overall endogenous function of the channel. So this work would not be possible, obviously, and I do this not only by myself. I have a good group of people that work on this project, and Taylor and Ruth were the two student and postdoc drove this project forward. Would not be possible again without the help of the I guess the resources that we have. Do I have a minute or two or not? Let's see.
You have probably 3 or 4 minutes.
Okay, so I wanted to share with you something that actually fascinates me, and we sort of had a conversation last night about the CBD. So CBD is quite an interesting compound, and some of us know that it's actually, for some reason, had a bad popularity contest before, considering because everybody wanted to go for THC. And of course, we know the cannabidiol does not have anything to do with your, I guess, good effect when people, smoke marijuana. And of course, in New York Times, the CBD got the splash with the big article saying that this is probably the drug or compound for everything on Earth, and that green teddy bear, I mean gummy bear, I think, represents everything when you go everywhere and you see that everything now infused with CBD. And FDA approved in 2018 CBD for the kids with the severe type of epilepsy. So TRP channels is quite known, sort of non-canonical, I think, cannabidiols activates those channels, and other natural compounds also activates those channels. But what got our attention is that one of the channels called TRPV2, what actually studied the least out of the pretty much all the TRPVs, is mostly activates by cannabidiol. And actually, Jensen in 2018 was able to show that CBD possibly the most potent activator for all of the TRP channels, and TRPV2 was one of them. And we are interested in TRPV2 from the point of view of cancer and cell outgrowth and metastasis. But of course, using Cryo-EM, we asked the question of the CBD activation of the channel, and can we see the CBD binding site. And so what we did, we pretty much discovered absolutely novel CBD binding site in a channel. It actually binds very closer to the membrane, but to our disappointment actually was that the channel wasn't open as wide as we wanted. So we went to the single channel recordings and we figured out that seems like CBD only partially activates the channel, so our partially resolved structure makes sense. The bottom of the gate is open, but the top is not, while CBD is fully wedged into the new binding pocket. The interesting part of the TRP channels, especially the TRPV1, I think, and TRPV2, is that their pore and their, what we call it, turret domain, I think, does mimics of opening and closing the channel even without the drugs there. So we'll be able to do the apostate of the channel. But from all this work, as you could imagine, now we have a new CBD binding site in the TRP channel, and we're going to try to do the same sort of story of trying to find the novel compounds and hopefully try to figure out how this channel could be fully open with the sort of structural drug by design. So right now the channel is only partially open. You could see here that it opens, but it must be needs another modulator. So, and again, here this everything actually was done in collaboration with Pfizer. And I have if you have questions, I'm finished. I wanted to show this video while people could ask the questions, because we collaborated with the Janet Isawa, who actually graciously made us a video about TRP channels and kidney disease. So while you're watching and thinking of the question, thank you.
So we have time for two or three, four questions. Maybe I can start with a quick question then. Therapeutically, I thought the effect of cannabinoids was more on GABAergic transmission.
So somehow these days, cannabinoid, we don't know what TRP channel is doing in nervous system. We really don't. And so somehow we see that the CBD does something to the TRPV subfamily for sure. But
if you were to knock out that channel and then look at the behavioral consequences of cannabinoids?
So nobody looked. So something that we actually want to explore, because seems like it has some interesting consequences.
Very nice presentation. For the compound that blocked TRPV5, but not TRPV6, is there a difference in the binding pocket between those two that explains that difference in pharmacology?
So it does actually. So it's a loop there that connects S3 and S4 that actually very well defined in TRPV5 channel and completely non-defined in any of the structures of TRPV6. So we think actually this loop that connects S5 and S4 somehow makes our pocket more tighter, allowing the drug going in, and does not allow that for TRPV6. So sequence-wise, though, over there is, they're 99% similar.
Maybe I can ask another question.
Yeah, go ahead.
Calmodulin effect, is that like a pore blocker? Is it like the N-type?
Yes, it's pretty much like a pore blocker, so it just comes in and just blocks the pore.
Is it voltage sensitive though?
Chips are not voltage very.
The calmodulin?
I actually don't know that answer. Huh. Thank you.
Any more questions from the audience? Anybody interested? Coming from Canada, we're all interested. habit of doing that, right? It's legalized there. So maybe if this was in Canada, there'd be plenty of